Okay, 3 weeks ago, I went for my booster jab to continue my protection against COVID. I'm one of the lucky majority. But for the vulnerable minority who are not protected by vaccines, there is an ongoing and urgent need for non-immunological treatments that can be used to prevent COVID infection. Today, I'm going to discuss the opportunity to repurpose the Japanese intravenous anticoagulant drug, Nafamostat, as an inhaled drug to prevent COVID infection. Early in the pandemic, Nafamostat was identified as the most potent inhibitor of an epithelial cell membrane protease enzyme called TMPRSS2. Now, COVID exploits TMPRSS2 to enter epithelial cells to start the infection cascade. In step 1, virus particles are inhaled, or they're transferred to the oral or nasal epithelium from contaminated hands and fingers. And at step 2, TMPRSS2 gives the virus particles a haircut. Spike proteins are cut and receptor binding domains are exposed. At step 3, viral particles bind to ACE2 receptors on the epithelial surface and then enter the host cell by endocytosis. Nafamostat can stop this process by inhibiting TMPRSS2 so that there is no haircut, and steps 2 and 3 are prevented from happening. And although Nafamostat is currently used as an intravenous drug, repurposing it for delivery by inhalation is the obvious and the most effective way to deliver it directly to the required site of action, the airway surface, where it can inhibit TMPRSS2. TMPRSS2 is a member of a large family of serine proteases that have evolved by gene replication. They perform many different biological functions, but the important thing to note is that the active site for this enzyme family is highly conserved. Nafamostat is a broad spectrum inhibitor across this protease family. The mechanism of action of the drug on TMPRSS2 is to rapidly acylate a serine amino acid in the active site. A stable enzyme inhibitor complex is formed, and the half-life of this complex is over 14 hours. The lifetime of the complex is therefore longer than the life of an enzyme protein in the epithelial membrane. And this means that the turnover time of the enzyme in the cell membrane is what controls the effective duration of TMPRSS2 inhibition. It also thus controls the duration of protection from COVID. We calculate that 4 times daily inhalation of Nafamostat will provide round-the-clock protection from COVID. To provide this protection, how much Nafamostat will need to be inhaled? We calculate that a 50 microgram dose will achieve maximum inhibition of TMPRSS2 in the airways, mouth, and nasal epithelium. 50 micrograms is 1/200th to 1/1000th of the doses that are used in the IV products. But is reducing the product dose size the only thing that we need to do for inhalation use? Unfortunately not. Translation of the Japanese Summary of Product Characteristics reveals that the IV formulation contains not only Nafamostat, but also 10% by weight of succinic acid. This acid content is totally unsuitable for inhalation. We will need to develop acid-free formulations for inhaled clinical use. But this should not be a problem, since acid-free non-clinical Nafamostat is widely available at biochemical reagent suppliers for use as a protease inhibitor in the biosciences. In conclusion, I've shown you briefly why and how Nafamostat should be repurposed to prevent COVID infection in the vulnerable. Our next steps are to seek and secure funding, support, and partners for development of an acid-free clinical formulation, for conducting preclinical inhalation safety and toxicity studies, and for early-stage clinical development. Thank you.